Hello, good evening to everyone who is with me on this webinar, as well as some of the invited guests temporarily on YouTube. Thank you for joining us as we go through this very interesting and complex topic. I've spent many hours sitting down, preparing the presentation, reflecting on the findings, trying to put everything together in a coherent way that is going to make sense. As usual, the first part of the presentation will be open, but beyond that, only people who had registered for this webinar will be with me. And so you can look out for the full details after this has been completed. And so we will start really at the beginning, and I will try and take you through quite a number of slides to help you to try and understand what it is that I am seeing in the context of the pandemic and what we need to expect as to being possible to occur in the near future. I'm just getting my slides prepared, and then we will start. But just to let you all know that when you reflect on the composition of the clots, it is very, very unusual. And we do need to think carefully about what could be the mechanisms in order to make sense of it. So I'm having a little bit of trouble loading up my slides, and I'm not quite sure why. Um, I'll try one more time. Yes, so as I was saying that this is a complex topic and something that requires a lot of thought and much further investigation. So this is just the starting point, and the purpose why I will continue to raise these points is because I recognize that it is critical for us to be able to grasp the significance of what is going on with regards to clots. What's important to take home at the end of this is that in reality, the mechanism that I'm going to be focused on, I think can happen um, over time, but it tends to be relatively quick because one of the things that I had received, one of the pieces of information I had received from somebody who reached out is that in reality, when the standard investigations are done, the radiologists and so on, they tend not to see much. And that suggests that the mechanism is slightly different in terms of the well patient. That's a very, very important point that we have to try and see if we can clarify. So, again, I just had some technical difficulties. If you just be patient with me as I try and pull together the slideshow so that we can take it from the beginning. Um, yeah, this is a good point from one of the webinar guests. I'd spoken to Professor Doug Kell from the University of Liverpool about microclots. Yes, my hope is to try and bring in some of the experts, share with them the information, and then try and understand from their perspective what they think the mechanisms could be. And that's something that definitely needs to happen so that we have as many thoughts and eyes on the concept. What's important for people to take away is that this is not conspiracy, and so don't allow anybody to put it away as a conspiracy theory. It's not at all. It's very unusual, and it is something that needs further investigation. I genuinely can't accept that the system has not acknowledged the importance of this, and therefore it needs further investigation. So I finally got my slides loaded. As I said, I'm having a few technical difficulties, so just be patient with me if I have a few problems. So let's start at the beginning. This is a composition and potential cause of these embalmers clots. This is really what I'm focused on at the moment. And as usual, there is always a disclaimer. This is information. It's not medical information in the sense that you should take this in place of seeing your doctor. If you have emergencies, if you are concerned, you must contact your physician or doctor to get treatment. This is just for the purposes of information that you can understand more about the science. The principle that I go for is that there is nothing new in medicine. And that was my first thought with regards to the abnormal clots. This is unlikely to be something new. This is just something different that needs to be understood. And so that was my starting point to try and understand where else this has happened and what does it mean. So again, this is what the key learning objectives will be: an overview of clots and the embalming process, talking about clots and the contribution of the spike protein, review of the actual constituents of the clots, and the likely explanation of what I call white clot syndrome. This is the fundamental as to what's going on. So this represents an extremely long embalmer's clot. So this is a picture of Richard Hirschman. He was one of the early embalmers who spoke up about this. He's holding a 19-inch clot here in a tube. You can see the whole white clot here. This is very clearly very, very abnormal. And as you can imagine, it's no surprise it's with an embalmer, because this is not really conducive to not causing serious complications in the person who had these clots in them. What's not clear is what was the cause of death for this person who had this kind of clot in them. Was it a coagulation thing or was it something else? And then I have here just an overview of some simple principles about blood and the constituents of blood. This is an example of whole blood. So this is what we would collect from you if we took some blood out of you. And then if we put it in a centrifuge where it was spun down, you would have the red blood cells, the erythrocytes at the bottom. Then you have the buffy coat, which would tend to have the white blood cells and platelets. It tends to look white. And then the plasma used a yellow color, which would have the proteins, the nutrients, metabolic waste, electrolytes. That's 55% of the total volume of blood. And in the context of the white clots, it seems as though primarily it's occurring with regards to plasma and not the red blood cells that would make it red in color. It's a little bit more complex than that because these are involved, but in slightly a different way. This here is just a reminder of the overview of the clotting pathway. And you tend to have an intrinsic pathway and an extrinsic pathway. Both pathways come together to cause the same thing, which is the triggering of fibrinogen into fibrin, which stabilizes these clots, which are then controlled by plasmin, which breaks them up. So everything in the body is about balance. So these pathways will trigger clot formation. Usually, say tissue factors, if there's a cut and there's exposure of the subendothelial region, it triggers the tissue factor, which would then cause clotting. Intrinsic pathways, a different mechanism. And you can see here warfarin is on both sides. And then some of the other more common, the 10A inhibitors, the 2A inhibitors, all of these are blood thinners that we use to try and control clotting when there are disease patterns. And I mention here again immunoglobulins, main types, IgM, that's the early immunoglobulin. It's essentially like the single immunoglobulin, the IgG, with 2 points, but it's all hinged together to make it into a pentamer. So 10 points where it can bind. It tends to be the first antibody that's produced in an infection. And then the long-term antibody is IgG. And then you have IgA, which can be in the bloodstream or on mucosal surfaces. This is what the body uses to defend the surfaces. I've mentioned this because I'm building the pieces that are involved in these embalmers clots. Then I'm showing here just a simple stable clot. So this is what a normal clot would look like, where the fibrin, as I mentioned, is linked together and it is usually quite dark. There's no stringy material in it. And this is just a normal clot. If you got cut or if you're coughing up some blood, it would tend to just look like this. And so it's important to understand what is normal from what is abnormal. Now, I've mentioned here the embalmer, and it's important to understand that what embalmers do are unique. Nobody else does what they do. So if embalmers are seeing something abnormal, there is no logical reason to think anybody else will see what they are seeing. So, really simple but important point. Doesn't matter how expert you are at bees, but if you study it in the lab and in university, what a beekeeper sees when he's handling bees is very different from what you may learn in a textbook. So we have to consider embalmers are at the front line and they are doing a very specific kind of work. And so this is what I'm highlighting here. So they are professionals. They prepare bodies after death. They're usually further trained than a mortician that's at a funeral home, and they have to adhere to very strict health and safety regulations. And essentially, they're injecting chemicals into the bloodstream to slow the natural decomposition of the body. And that's what their job is. And it's really so that they can preserve bodies for longer periods of time. If there's a delay, for instance, with regards to funeral. So if you're not being buried immediately, you may need to use this in order to protect the body. And the procedure, they use a pump and they tend to go at two locations. This is up by the neck or in the femoral region. They pump the fluid in, and as they pump the fluid in, the blood then comes out. And it's the fluid that goes and circulates through the body that then prevents the decomposition. And so this is a very important point. What they're doing here is not done at autopsy. So when you're doing an autopsy, you are cutting the body and you may look at specific vessels, but you are certainly not doing what an embalmer does, which is to flush out the blood out of the blood vessels. And so expecting pathologists to see this pattern is not, in my mind, practical. It would only occur in people who are doing that specific job. And so this is why it is so inconceivable that even if one or two out of thousands were noticing something different, it should be investigated. But we're talking about hundreds of them as saying this is happening, and here is the reality, it does affect their work because every time the system clogs up, it takes them longer to therefore finish a body. So it's having direct impact on their costs and their overheads. So to discount it, I mean, you can say you don't know, but you don't discount it. You don't say that it's a conspiracy. It is what it is. Whether or not it's relevant in terms of the cause may not necessarily be so easy to clarify. But as I said, the first stage is to just acknowledge that it's occurring, and then it's a question as to should it be further investigated. That's a different part of the question. Here is the early part of the breakdown of what these abnormal looking 19 up to 19-inch clots had in them. And what I did is I broke down into normal in black, blue I've put as kind of in between, and red not really present in normal clots. And you can see of all of these proteins here, how many that are red are present. You know, band 3 and ion transpoint protein, alpha 2 macroglobulin, you know, immunoglobulin heavy constant gamma 2. But compared to the fibrinogens, the fibronectins, the integrin, the vitronectin in black, these are normal. But all of this red stuff, why is it becoming caught up in these clots? And that's really part of the question. And what I am trying to do is I'm trying to see if I can make logical sense from a scientific point of view as to what exactly is in or triggering these abnormal clots from occurring. And as I said here, in terms of died suddenly, this is the one that triggered the interest. It was a program, kind of documentary kind of thing, with Stu Peters. But at the time, I think what happened is that it was perceived as an anti-vax conspiracy. And you can see on this glove here, the white is these abnormal clots, which are very elastic and very difficult to break down. And the dark red here would be more like a normal clot, and they're mixed together. And so, what the embalmers are seeing are lots more of these white clots, primarily since 2021, but they did see them in 2020 as well. So, there is likely to be a combination of factors that's causing it. But the fact that it was labeled as a conspiracy is probably the main reason that it wasn't thoroughly investigated. I'm not sure if that's acceptable. I can understand the thinking from the scientific community, but you should still investigate what is actually happening. Where I got really, really seriously focused on it was when a whistleblower spoke to me, and he reached out and said, these are not just occurring with the dead, because you have to, for anybody who's seen my initial thoughts with regards to the cause of these clots, I thought these were more cryoglobulins, because I was looking for abnormal patterns that occurred after death when the body cools. If there were high amounts of immunoglobulins in the bloodstream, they could cool and then form these clumps, which in my view could have caused these abnormal clots. But when somebody reached out with images that would demonstrate that this is being pulled out of living people, it confirmed what the embalmers were saying, because the embalmers said, we think this is occurring prior to death because we have had bodies within an hour of death and we're finding them. And at the time, I thought, well, that doesn't make sense. We should have seen this clinically. But when you have somebody then show you direct evidence of this occurring, then that means it should not definitely be ignored. And this is exactly the kind of thing that they have been removing from living people. And so, what I've shown here is that this line here represents what would be the lining of the inferior vena cava, and this would be an example of where this clot was sitting. It was pulled out, so the person survived. The red part of the clots are here, but these white clots are also mixed in with it. And this is the kind of thing that seems to be in existence. Very, very serious, and not to be therefore underestimated or ignored. So when I saw that, I think that I had a responsibility to continue to talk about it and to raise awareness. And so what I'm doing is continuing to raise awareness of it until it is thoroughly investigated by the scientific community. And as I said, in medicine, there is nothing new. It's just refashioned. I'm hoping that by the end of the presentation, those people who are with me on the webinar will understand why I said that this is a refashioned disease, in the sense that it does happen, but very rarely. And there is something unusual that is occurring in and around the pandemic that could be triggering the formation of these events. As I said, this is my theory, and I'll be sharing to the webinar registrants what is the theory from the document about exactly what the cause of the mechanism is. And so before we take off the YouTube cohort, just in case you hadn't seen this, this is what I am talking about here. The most recent top secret solution of Scientists International. And what they have done, the authors are anonymous because they are concerned about their safety and their profession. But they have then taken a number of these clots and they have analyzed them, and they've broken it down. And you can see this is quite a long document, which I'll be going through. But this is the part here where they took 4 different clots, one in a young teenage female, one in a 25-year-old male, another one in a 40-year-old unknown gender, and a fourth one in a 70-year-old. And then they broke it down into the constituent parts. So part of my presentation will be to look at their theory. And so they have in here their own perspective, with regards to fibrin and fibrinogen. And then my part of the presentation will be to try and add on to that why I think that this is likely to be um, a combination of factors as to the actual cause. So thank you if you're on YouTube and you've joined us, but I will let you go in just a minute. But just to highlight the point, remember this: this is not normal and it really needs to be investigated. Don't let anyone get away with saying that it is a conspiracy. They can say that they don't understand it, they've not seen it before, but it absolutely needs to be investigated thoroughly. So for the registrants on the webinar, just hold on while we let our YouTube audience go.